All right. Good evening, everyone, and welcome to the latest event in the Blue Space Angler event series, which is Nutrient Management, Cyanobacteria, and Fish Health. In this presentation, we're going to be talking about how different lake cycles are affecting the health of our fish population. So this presentation is brought to you tonight by the City of Worcester and the Indian Lake Watershed Association. My name is Jacqueline Burmeister, and I coordinate the Lakes and Ponds program. I'm here with the president of the Indian Lake Watershed Association, and I'm really excited about our special guests tonight, Jason Stellarski and Nancy Leland, who are going to be sharing on these topics. So as I mentioned, this program is part of the 2021 Angler Event Series, which, as its name suggests, is a series of 13 events that pertains to all things water quality, fish, and fishing. It's a collaboration with our local watershed associations to engage new and existing anglers in water quality challenges around the city and promote the great fishing that is occurring in our lakes and ponds and everything that they have to offer. So if you like this presentation, head on over to wooanglerseries.com where you probably registered for this one. There are, I think, like 7 or 8 more this year that are coming up. So you should check them out. So before we begin, you may or may not have heard the word cyanobacteria in the past, but you're going to hear it a lot tonight. So I wanted to give you some basic definitions. So cyanobacteria are also referred to sometimes as blue-green algae. You might have heard that term. They're simple, single-celled organisms that could live in colonies such as these here on the screen below. They are fascinating. They're the oldest form of life on Earth, being responsible for the oxygenation of our atmosphere 2.4 billion years ago. So they're responsible for the evolution of those higher forms of life and us. So today they are still very resilient, and they're found in almost every ecosystem on the planet, from the Arctic to the deserts to the oceans and our local lakes and ponds. And so in a healthy lake, they're present in low numbers. They use sunlight and carbon dioxide to photosynthesize. And that's that process that we learned about back in biology, where they take oxygen or they take carbon dioxide and use sunlight to create oxygen in the water column. And so that's great and fine, but under the right conditions, they can explode in population, creating what are known as harmful cyanobacteria blooms. And this is when you see the lake turn a color of green or see this big film of mat on top of it, and it's kind of gross. This is a picture here of a bloom that we had on Coe's Reservoir back in October of 2019. So these blooms are harmful for several reasons. One is that when they die, they cause low oxygen conditions that are bad for fish. Another is that they could produce toxins that are harmful to humans and animals. And so we're still learning to understand the factors that contribute to harmful cyanobacteria blooms, but we do understand that they really like nutrients such as phosphorus and nitrogen and warmer waters. And so blooms are becoming more of a common occurrence in our region, and they have already affected Worcester. Indian Lake, for example, has been troubled by cyanobacteria in the past. Beth, would you like to talk a little bit about Indian Lake and the situation that we have there?
Good evening, everyone. I want to thank you all for being here, and thank you, Jacqueline. Again, the Indian Lake Watershed Association, I'm president of the group, and I have been for probably about 5 or 6 years now. We are a nonprofit that was established nearly 40 years ago by local residents to protect and preserve Indian Lake. While this is still our primary purpose, we also assist with maintenance at public spaces around the lake, 2 neighborhood war memorials, and are involved in various other projects to enhance the neighborhood surrounding Indian Lake. At just over 190 acres, Indian Lake, located in the northern part of Worcester, is the largest water body located entirely in the city of Worcester. I want to move on to our next. Previous to the establishment of the City of Worcester Lakes and Ponds program, Indian Lake experienced a serious cyanobacteria bloom in 2014. As a shallow, warm water body, it was not uncommon for the lake to experience algae blooms. Indian Lake also has a lot of bottom-feeding fish, which tend to stir up the bottom sediments. Therefore, the lake has never had optimal water optimal water clarity. There are hundreds of different types of algae that occur naturally, but cyanobacteria, also known as blue-green algae, can multiply very quickly under the right conditions and can produce toxins that can be dangerous to humans and pets. At the time the lake was in full bloom, it had a bright green tint, although not quite as bright as this photo. This picture was actually taken at a small private lake in Holden at the time of our bloom. The resident was afraid that whatever was affecting her lake was something that was also affecting Indian Lake. Indian Lake was closed due to the high cyanobacteria count in early July of 2014, as that count was determined to be beyond the state threshold. Because conditions can change so rapidly and cyanobacteria has the potential to produce harmful toxins, the state of Massachusetts has a very low threshold for cyanobacteria. When a water body reaches this threshold, an advisory is issued warning people not to swim. While we have learned a tremendous amount since that time about the challenges with cyanobacteria, the scariest piece is the second photo. Toxins were not present during our closure until late September, when the lake looked completely normal. The lake did not reopen to the public again until October of 2014. Our organization and our counterparts across the city at Lake Quinsigamon and Tatnick Brook Watershed Associations have advocated for years for our respective waterways, but this really brought everything to a whole new level. The increase in cyanobacteria blooms across the country is a serious threat to public health. We are grateful to the City of Worcester for establishing the Lakes and Ponds program to work with our groups to develop short and long-range plans for our recreational waterways. The City of Worcester is one of only a handful of cities and towns that monitor recreational waterways for harmful algae blooms, which you will learn more about that tonight. It is so important. Collaborations such as tonight's program and the Worcester Angler Series help to educate and bring more people to the table to appreciate the value of our blue spaces. Thank you very much.
Thanks, Beth. So with that, I'm going to hand it off to our guests here tonight. So first up, we have Jason Stellarski, who is from Mass Wildlife, and he's going to be talking to us about how cyanobacteria fit into the natural lake life cycle and how external factors are causing blooms to become more frequent. And after that, we're going to have Nancy Leland from the University of New Hampshire, who's going to talk about toxins that are produced by those cyanobacteria and how they move through the food web and perhaps ultimately to our fish. There's going to be an opportunity to ask questions in between those presentations. And so you'll be able to unmute yourself or type a question into the chat box for our presenters. Before we hand it off, do we have any questions up to this point? Open the question box. I don't see any in the chat. So, Jason, I'm going to hand it off to you.
Great. As Jacqueline said, my name is Jason Stellarski. I work at Mass Wildlife. I'd first like to thank everyone for inviting me to speak at this event. I think it's a great idea, the citizen science, the pairing between city government and general people and lake associations. It's excellent. You don't see it that often in the state, and it's refreshing. So tonight I'm going to be talking a little bit about water temperature and oxygen mainly, and acute effects to fish. I don't know everyone's background who's listening to this talk, but I decided to go kind of basic tonight. But feel free after the talk to answer or to ask me any maybe more specific questions. But first, I need a shameless plug to Mass Wildlife. We're responsible for the conservation of freshwater fish and wildlife in the Commonwealth. We work to restore, protect, and manage land for wildlife to thrive and for people to enjoy. We handle hunting, fishing, wildlife. We have 5 district offices throughout the state. Normally, when I give a presentation, I'm actually physically speaking to a group of people at an actual place. So I would identify the local district office to them, but because everyone is virtual now, I don't know exactly where you're tuning into this presentation. So the central district office is the one that is responsible for Worcester. It's on Templeton Road. It's closed now currently, but when they open back up again, you can feel free to drop in anytime. They're all really nice over there. We also have 5 hatcheries for which we produce 500,000 pounds of trout annually. And we're just at the tail end of the stocking season. And if you are a trout fisherman, you can go to mass.gov/trout to find complete up-to-date listings of where the fish are and how to get them. So starting broad, lakes in Massachusetts. Massachusetts contains just under 3,000 named lakes and ponds. It's quite a lot. They're primarily formed via dams. They're reservoirs, and with a small number of kettle hole ponds or natural ponds, which primarily are out on the Cape where there's lots of sand and gravel. Now, these reservoirs are dams on small streams and rivers, and were primarily driven by industry in the 1800s, and early 1900s to use for power. And in fact, the deterioration of these dams over the past 30 to 40 years is leading to numerous removals every single year. Our sister agency, Department of Ecological Restoration, I think has 5 or 6 slated for this year for removal. And in Massachusetts, lakes are primarily used for drinking water supply. There's numerous reservoirs, including Quabbin and Wachusett, which are the 2 largest lakes in the state. For power generation, also for recreation and just aesthetics. As you can all probably agree, lakes are very, very pretty, aesthetically pleasing places. And now the bad. Massachusetts is the 3rd most densely populated state in the Union. This right here is an image of it's taken from space, obviously. The black areas are areas that are generally forested, whereas the pink to purple areas are areas that are highly covered with impervious cover. So they're hardened surfaces, either buildings or roads or parking lots. And it's amazing when you look at this picture, what you can discern from a simple image. I mean, I don't know if you can see my cursor, but you can see Springfield. Big area of Springfield. You can see Worcester to the east, and if you go north, you can see Leominster, and move to the west is Fitchburg, and then of course the big area you see here is Boston, which is almost completely hardened. And these surfaces create problems for water. They prevent water from seeping into the forest, into the land, and they intercept that water and divert it to streams. So human modification of these landscapes alters flow pathways of water. Water no longer goes into groundwater and enters streams through groundwater recharge. A lot of water now is directly run off into streams. And when that occurs, sometimes that runoff can have greater amounts of nutrients in it, nutrients that aren't absorbed in the soil as they otherwise would be if the water was to go through the traditional transport to streams, which is through the soil. And when runoff carries this elevated nutrient concentration, streams direct it right to lakes and ponds, because remember, a lot of the water bodies in Massachusetts are dams on rivers and streams. So if a nutrient-laden runoff makes it into a stream, it's eventually going to be stopped in an impoundment. And these impoundments act as nutrient sinks. And this has been occurring for hundreds of years. And it contributes to lake aging. Now, lake aging is a natural process that's accelerated by human activity. And here's the spectrum, just kind of broad classifications that people like to use. On the left-hand side, we have oligotrophic lakes, which are generally free of sedimentation. They generally have clearer water, few aquatic vegetation. They're not much aquatic vegetation. And then we transition to a mesotrophic lake, which is somewhat impacted by nutrients. It tends to have a larger amount of sediment built up at the bottom, that muck you can step in when you step into a lake sometimes. It has more aquatic vegetation, water clarity is somewhat impaired, and they have some algae during the summer. And then full on to eutrophic lakes, which are largely filled with sediment, have murky water, are choked with aquatic vegetation. And these lakes and ponds, even in Massachusetts, lie on a spectrum. If you've ever been to Quabbin, you know that it's a very pristine area, and they keep it pristine because they've owned, DCR owns much of the landscape around Quabbin, and they keep it in that natural context, which allows any nutrients on the surface to be absorbed by the watershed and thus intercepted from entering into the water body proper. And then there are other examples of eutrophic ponds that I'm sure you know of in your travels throughout the state. So this lake aging is also called eutrophication. You're all very familiar with it. Some of its symptoms are anoxia, so areas where there's areas of the water column that are devoid of oxygen. This includes some of the other symptoms: fine sediment accumulation. If you've ever walked into a pond, and I have a number of times, and you step into muck past your knee, and this can contribute to decreased water transparency, more frequent algal blooms, which Nancy is going to talk about, increased vegetative cover. In both those two prior bullets, algal blooms and vegetative cover can contribute to greater amounts of anoxic habitat throughout the summer, because when algae dies or when the plants die, they settle to the bottom and they are decomposed. In those processes, that decomposition consumes oxygen. Now, all these symptoms of eutrophication can influence fish over the long run. Now, when I was putting this presentation together, I was thinking to myself, how detailed do I want to get? Because every single pond and every single species will react differently to eutrophication. Some ponds are more resistant to the effects of eutrophication, say kettle hole ponds. Most kettle hole ponds don't have tributaries that enter them. The water is exchanged through groundwater primarily due to the high proportions of sand and gravel out on the Cape. They still eutrophy, but at slower rates. And some species are naturally resistant to eutrophication, such like sunfish, which prefer littoral habitats that happen to be very weedy. So I decided to kind of bin the species into broader guilds and just focus on warm water fish and cold water fish. Warm water fish generally include bass, largemouth bass, smallmouth bass, sunfish. So I don't know how familiar everyone is with fish species: chain pickerel, yellow perch, white perch, and brown and yellow bullhead. And it's interesting that, when you have a slightly eutrophied system, generally, this can result in greater abundance and growth of warm water fish. And if you think about the theory behind that, it makes sense. You have excess nutrients in the water column. This allows for more autotrophic growth, so more organisms that use sunlight to produce energy, which, more algae, more phytoplankton, which are consumed by zooplankton. You get more zooplankton, more aquatic invertebrates, which consume the zooplankton, and then up and up and up and up until you get to the highest consumers, the large predatory fish. So excess nutrients can fuel greater abundance and growth of warm water fish. And the caveat is, and that's to a point. To a point, every species will become affected by eutrophication. And as I said before, that point is pond and species specific. And those points are we generally refer to as negative feedback. So the excess nutrients, are beneficial to a point until there are negative feedback loops which come back into play and then depress growth and abundance. And some of these examples of negative feedback are habitat homogenization. So when you have a heavily eutrophied system, there's not a lot of different habitat types. So the niche space, the available habitats for fish to kind of partition themselves and live their lives becomes smaller. And this can be due to sedimentation. So certain fish like yellow perch like to forage on long sandy banks because they feed off of invertebrates that bury themselves in the sand. If that sandy area is completely covered over with muck, then the yellow perch don't have that habitat type to use. And the same could be said for aquatic vegetation. If the entire 100% area of the pond is one monoculture of an excessive aquatic plant, there isn't much variability for the fish to take advantage of. Also, decreases in water clarity can affect foraging success. You can imagine in a pond that has very low water clarity, fish can't see very far. It's more difficult for them to find and catch prey. And a major limiting factor or a major negative feedback loop is this anoxic zone that develops throughout the summer. And it physically limits where a fish can go, because fish obviously need to breathe oxygen. And if there isn't any in a particular area of the water column, those fish cannot traverse those areas. And this can force fish into suboptimal habitats, which can cause stress and mortality if it's long-lived. So once again, when I was putting this talk together, I didn't know exactly how much everyone was kind of up to speed on kind of limnology. So I just decided to put a few figures together. So during the summer, we typically see higher oxygen at the surface and lower oxygen at the bottom. Obviously, you know, the sun can penetrate through the water column to a certain period or to a certain point. Autotrophic organisms, plants, and algae accept that or intercept that light and turn it into oxygen. They consume carbon dioxide, they emit oxygen, which is why you generally have more oxygen at the surface. Now, at the bottom, there's lots of decomposition that's going on, and these are oxygen consuming processes. And as the season progresses, these low areas of the water, the deeper depths, can go anoxic or devoid of oxygen. And that anoxic area grows throughout the summer. Typically, in May and June, it's very small or not apparent, and it grows throughout the summer. And density gradients caused by temperature changes in the water column kind of trap that anoxic area down in the depths and doesn't allow water to circulation. This permits the anoxic area to grow throughout the summer in July. And then in August and September is when we typically see acute effects of this anoxic zone on fish. And we don't see it everywhere, but it can crop up in certain years. Particularly if we have a fall heat wave. There was a few years ago, we had a pretty strong fall heat wave, and this stressed fish out because if you imagine fish, you know, they can't go any deeper than the anoxic zone because there's no oxygen, and it traps them in the very, you know, upper area of the water column. Now, if you get a really strong fall heat wave, this surface water can exceed the thermal thresholds of even warm water fish. I mean, these fish are temperate. You know, they evolved in temperate regions. They're not tropical like peacock bass or something, which can withstand very, very, very warm temperatures. So if you have very hot conditions late in the summer when the anoxic zone is very large, the fish really don't have anywhere to go, and it kind of pushes them into these suboptimal temperatures or habitats, and that can be physiologically stressful. It can lead to fungus outbreaks and disease outbreaks, and can even result in mortality if that anoxic zone envelops the surface waters. And this doesn't happen very often naturally in the summer. We do see it occur in the winter after long periods of ice and snow. It prevents light filtration into the water column, and slowly over the winter, the amount of oxygen slowly goes down and down and down. And if that ice holds on to late in the year, say to March, the water column can go completely devoid of oxygen, and that's really dependent upon how serious and how intense the winter is. There are also daily cycling events as well, and I'm not going to go into detail on those. Those are generally what you see in very swampy, marshy, shallow, very extremely eutrophic lakes or ponds. So for cold water fish, it kind of works in the same way, and anoxic zones kind of limit where cold water fish can go. So cold water fish, such as trout, the ones that we stock in the lakes and ponds, have certain habitat requirements. They need 5 milligrams per liter of oxygen, and temperatures have to be below 25 degrees Celsius, which is about 74, 75 degrees. So you can imagine as this anoxic area grows throughout the summer, the coldest water is on the bottom of the water column, the warmest water is on the top. And as that anoxic zone moves towards the surface, the fish becomes squeezed between warmer surface waters and anoxic bottom conditions. And just like with warm water fish, they get pushed into suboptimal temperatures. The difference here is that trout, require this cold temperatures. So the anoxic zone doesn't have to be very large before the trout get pushed into too warm of water. And once again, this can result in stress and mortality. A few years ago, there was a drought in 2016, a famous drought actually, and a few ponds we lost our cold water layer, and it resulted in large trout. Well, not large trout kills, but there was definitely a distinct signature of that. We knew exactly what happened, and it was unfortunate, but it does happen from time to time. So warm water fish can get stressed. Not mainly, but predominantly when we have warmer temperatures later in the season. But cold water fish become stressed very easily in every single year. There's my little diagram for trout water So after this, this kind of just basic information, eutrophication is a natural process that accelerated by humans. Normally it would take thousands of years for these processes to see themselves through, and it's turning into hundreds of years. I'm sure all of you, I know I can, I'm getting old. I remember lots of ponds that I used to recreate on as a kid that are now way more weedy than they used to be. So these processes are evident in a human lifetime. They are initially beneficial to warm water fish to a point. Cold water fish tend to be more susceptible than warm water fish, and every pond and species react in a different way. Some ponds have thermal refugia, springs, or cold water tributaries that cold water fish can take refuge in, thermal refuge. And kettle hole ponds, may be slightly less susceptible due to the large proportion of groundwater that they receive. And yeah, most detrimental to cold water fish. So that's all I really have or wanted to cover. But I would entertain more complex questions if anyone has any. Let me stop sharing here.
Excellent. Thank you, Jason. Does anyone have any questions? You could unmute yourself or you could type it into the chat box. When there are toxic blooms, how do you control them? That is a great question.
That's a Nancy question.
Yeah. We have a few things that we'll talk about what we do in the city of Worcester. And maybe Nancy could talk about it a little bit in her presentation.
Hello, everyone. I am Nancy Leland. I am an affiliate faculty at the University of New Hampshire, Center for Freshwater Biology and Ecotoxicology. And we focus just on cyanobacteria now. There was a day when the Center for Freshwater Biology would entertain working on most anything that was related to freshwater biology, streams and ponds and all sorts of different things. But now all we do is cyanobacteria. So one of my projects that I have been working on is working with fish populations. And that's the reason why Jackie invited me here tonight to talk to you about the research that I've been doing on fish health and evaluation of the interactions between fish and cyanobacterial populations. Next slide. So I'm going to be giving you an introduction to the cyanobacteria from my perspective, talking a little bit about the various exposure pathways of the cyanotoxins, and the cyanotoxins are those toxins that are produced by the cyanobacteria into aquatic food webs specifically. That does not mean that they don't transfer into terrestrial food webs, they do, but we're just going to be talking about the aquatic portion. And then I'm going to bring you down to Cape Cod to see what I've been doing down there for the last couple of years on the impact of Alewife on cyanobacterial populations in a particular site, Lower Mill Pond. And then talking about the results of that study in terms of the bioaccumulation of the cyanotoxins in these alewife. And then what about Worcester? Next slide, please. So the cyanobacterial populations are really quite interesting, and they are incredibly dynamic and unique by genus and even sometimes species. So it makes for a very complicated evaluation when you're looking at the data that you might be collecting. For example, if any of you are doing microscopic evaluation and looking at your images and submitting images for the Cyanoscope program, these are very unique populations, and they describe various exposure pathways for us as we look at them. We break them into two parts, basically. We look at what's called the Pico Cyanobacteria, which are very small cyanobacteria. They tend to be in single cell form. Sometimes they form into colonies, and that is genus specific, whether they do form colonies or not. But more often than not, we're dealing with them as single cell forms. And we also sort of lump them into what we call the edible fraction, because they are capable of being ingested and moved through the food web. And then there's the bloom-forming cyanobacteria, which we tend to consider as being the inedible forms. These are the forms that you'll tend to see if you take a sample and look at it under the microscope of a bloom or a scum material or some type of a surface accumulation along the shoreline. So as you can see in this slide, that they're all very different genus that we're looking at. So if you do an identification for Jackie and you're indicating to her what you're seeing, you're kind of letting her know whether you're seeing something that might be considered edible in my world, and then maybe something that is considered inedible. And they're very important distinctions between the two. Next. So the Pico Cyanobacteria, they are very planktonic, which means that they move around in the water column based upon what's happening with water circulation and potentially wind. And we at UNH tend to think of them in terms of problems with transparency in the water column. And that is in comparison to something that is buoyant, which is the bloom-forming cyanobacteria. And those are typically associated with the surface scums and the blooms that we see that when the state agencies say, you know, have you seen a scum? And that means that they say, oh, this is a bad condition, and we come out and we take a sample. So those are the BFCs. That's the inedible fraction. Next. And the interesting thing about these two different populations of the cyanobacteria, the Picos and the BFCs, is that we have different amounts of these cyanotoxins that they tend to produce. So there's many cyanotoxins that they produce, but we tend to focus on 3 of them with the research that we do at UNH. We focus on something called B-methylalanine, or BMAA. That is a cyanotoxin that is associated with ALS. There is also anatoxin A, which is associated more with acute exposure. And then there's the microcystins, which are the cyanotoxin that is currently being considered for regulation, where you'll see health advisories for recreational waters is for the microcystins. So the interesting thing about all of these toxins is that they are very highly water soluble. So we tend to see them not only in what we call particulate form, which is contained within the cells themselves, but also dissolved in the water column. And that creates some interesting things in terms of the potential exposure pathways for these different toxins, is whether they're in the particulate or dissolved form. Many times we will find that these toxins are co-occurring. So if you go into a system, a pond, and you collect a sample, what we do in the lab is we will evaluate for all 3 toxins because we typically find them there co-occurring at the same time. They have different modes of action specific to the type of toxin. BMAA is more of a neurotoxin, which is a chronic condition. Anatoxin A is a neurotoxin as well, but that is an acute toxin. If anybody is sort of paying attention to what's going on with anatoxin exposure, it is associated with these reported animal deaths that we've seen, dog deaths and cattle. So it's not anything to be messed around with, but very important acute neurotoxin. And then the microcystins are are a hepatotoxin, can be in the acute form in terms of exposure, but typically more of a long-term chronic exposure profile to that. And then, excuse me, the degradation of these toxins becomes very important. Of what happens in the natural system is how long do these toxins persist in either particulate or dissolved form when they're in the water body. So some of them are reported for being there for just hours, some for days, and some for weeks. So if you're thinking about exposure profiles, it becomes really important to have in the back of your mind is how long are these toxins sort of hanging around and being problematic, either for us as humans going in to potentially recreate or drink this water, or for what I look at, which is the food webs. And mentioning food webs, there's bioaccumulation. So that's what I'm working on now is what are we seeing in terms of bioaccumulation of these particular toxins as they're moving through the food web. Next. So some of the things that we don't know about cyanobacteria and cyanotoxins is after many years, it's been probably 30 years, believe it or not, that people have been researching and trying to understand what are the triggers for toxin production. And we still do not know exactly what the triggers are that can make what we call a toxigenic cyanobacteria start to produce the toxins themselves. We understand some of the environmental conditions that are optimal for these particular cyanobacteria, but really understanding the triggers, we do not know yet. So it remains a puzzle. We do not really understand the human health risks associated with the BMAA or the anotoxin. And we have a very limited understanding of overall what the ecological risks are associated with any of these toxins. Next slide. So how can cyanobacteria and their toxins that they produce affect the fish populations? We have the three different toxins. We have microcystins, the anotoxin, and the BMAA. We have our planktonic Pico Cyanobacteria, which are typically in single cell form. And then we also have our bloom-forming cyanobacteria, which are very buoyant, and as I mentioned, are the ones that tend to show up as surface accumulations. So in a sort of a simplified way of thinking about these bioaccumulation pathways that we have, we can see them accumulating in the zooplankton. So what happens with the zooplankton is that they are capable of ingesting these smaller forms, these planktonic Pico Cyanobacteria that are less than 50 microns in size. So we can sometimes see that toxin accumulating from the water column into the zooplankton. And we can also have bioaccumulation in the fish themselves. So that's a result of the food transfer as these fish are foraging around and eating whatever, feeding upon whatever they need to feed upon based upon their strategy. So they can be ingesting the zooplankton if they're planktivores. They can be ingesting crustaceans and mollusks and other panfish if they're piscivores. And then the fish can also be exposed through ingesting dissolved toxins across the fish gills, but that tends to be a much lower amount of exposure through the dissolved toxin pathway. Next. So as I mentioned, the zooplankton are typically the most important player in this whole game of the food web to see where these toxins are going. And the reason being is that they are ingesting different particle sizes within the water column of the cyanobacteria, typically less than 50 microns in size, as I mentioned. And then we can also have the edible Pico Cyanobacteria being ingested into the mollusks and the crustaceans that are living in the benthos. So if you have freshwater mollusks or crayfish, they will be exposed to these cyanotoxins as well. Next. So when we start thinking about what happens with the fish populations, we start breaking them into feeding strategies and thinking about how they might be exposed to the cyanotoxins. So we have our planktivores, which tend to be pelagic. And in that particular group, you could have something like your bluegills or your perch. Next. And then those are fed upon by the piscivores, which are also pelagic. So you have your bass and your pike. So now you have the toxins moving from typically what would be the planktonic Pico Cyanobacteria into the zooplankton, into your planktivores, and then into your piscivores. Next. And then we also have to think about our omnivores. So they're sort of benthopelagic in terms of their feeding strategy. They can move from feeding on something that's in the water column, and then they can also move into the benthos and feed upon things that may be living in the sediment or actually ingesting directly some of the cyanobacteria that are in the sediment layer. And there's actually a lot of the cyanobacteria that remain in the sediment every year seasonally. So they do not always, the whole population does not always migrate into the water column. There's always something in the sediment itself. So the sediment source is a very interesting thing to investigate and has a lot to do with what we're looking at in terms of the benthopelagic population of fish. Next. And then we have the detritivores, which are the benthic carp. So they're pretty much obligate in terms of feeding upon detritus that's in the benthos. And actually, sort of I'm giving away a secret that I'm going to be sharing with you in a couple of slides, but you know, they have, as you might think, have a very high body burden of these cyanotoxins because, you know, that's their feeding strategy, and they're feeding in the benthos, and there's a lot of cyanobacteria in the benthos, and they're down there sort of, you know, scurrying around and roughing things up and redistributing all of these cyanobacteria, and they're ingesting them. So it's a really interesting way of looking at what's happening with these different feeding strategies and how the fish come into contact with them. And there's always the process of sedimentation going on with these BFCs. There's the dissolved fraction, which is constantly changing through the season as we go along. So there's many different ways that the fish populations could be exposed to these cyanotoxins. Next slide. So I'm going to bring you down to Brewster, Mass, which is down on the Cape. And this is the Lower Mill Pond Alewife study that I've been working on for the past 2 years and is going to continue again this year. And we have been looking at the interactions between the juvenile alewife and the cyanobacterial populations in Lower Mill Pond. Next. So in Lower Mill Pond, what happens is they have an adult alewife in migration occurs early May and end of April, early May. The adults are coming in and they are spawning. And so we have these juveniles that are being hatched basically in Lower Mill Pond. And then by sort of the middle of July, they start their out migration. So we have been, and as you can see in this collection of the juvenile alewife in this net, is that they're really, really tiny. They're just so small, 40 millimeters, 50 millimeters, really tiny specimens when we collect them when they're out migrating. But one of the more interesting things about what we saw for bioaccumulation of the cyanotoxins in these juvenile alewife is we don't know when the bioaccumulation of the cyanotoxins begins in this 10-week period between the in-migration of the adults and when we collect them during the out-migration in the middle of July. Next. So I want to share with you what we found for what was happening in Lower Mill Pond in Worcester. So this is more of a seasonal trend of what was happening with the cyanobacterial populations and the zooplankton and the fish in Lower Mill Pond. So in early May, we had what's called a really diverse population of zooplankton in Lower Mill Pond. And that tends to represent a system that is in balance in terms of what's happening with predation and nutrient levels. So it's considered to be a healthy zooplankton population at that time in May. Next. And then the juvenile alewife are hatched. And so they are planktivorous when they're young. And their preferred food is the Daphnia, which is you see on your far left side, the big guy that's got his arms up. That's their preferred food source, and they will start eating the Daphnia first. And that tends to shift the zooplankton population from something that is, as you can see, is relatively diverse into something that is not quite so diverse. Next. And that's exactly what we observed, is that the juvenile alewife basically eliminated the Daphnia from the system, and they also eliminated the copepods from the system. And we were left with what are called the smaller crustaceans, which are the Bosmina that you see on the bottom there, 20 microns. That's their feeding source in particle size, Bosmina, and then the rotifers, which feed on something that are about 2 microns in size. Next. And then by the June, all we have in Lower Mill Pond are rotifers, and they are preferentially feeding on things that are 2 microns in size. So this whole world of the feeding strategy of the zooplankton is all within the edible fraction. And you can see that we've focused ourselves into just feeding on something that's 2 microns in size. Next. And so what that means is that they're feeding on the Pico Cyanobacteria, these really small planktonic forms. And that eventually becomes an issue for Lower Mill Pond, which I'll talk about in a minute. But the thing that's more interesting, next, please, is that the juvenile alewife switch from something that's called planktivory to detritivory, which means they're changing from feeding in the open water column and then feeding in the benthos. And so this becomes really, really interesting because now they've changed their feeding strategy. And so now they have also changed their exposure pathway. From something that I would look at and say, okay, if they were planktivorous, they'd be feeding on the zooplankton. And so that's how I would want to do my analysis of what their exposure potential is, to something like, oh, they're feeding in the sediment. What's in the sediment layer? And that brings in a whole different, it kind of like melds the world of the Pico Cyanobacteria that were in the water column to something like the BFCs that were then, sinking in the fall and becoming more on the sediment layer. So it really created this amazing matrix in terms of us to figure out what was happening with these alewife and how are they getting exposed and what does that mean for this particular fish population. Next. So one of the things that we haven't quite identified, but is very important to what's going on, particularly with these alewife, is that what is it that initiates the change in their feeding strategy? One of the possibilities could be an age-related thing, and that is typically referred to as an ontogenetic response, or whether it's something that they drive themselves in terms of needing their food and changing to a feeding strategy that puts them into feeding on the benthos and more exposure to cyanobacteria. We really don't know. I would say, you know, in the literature, there's very few studies that have been done on this, but it's kind of like, well, we really don't know. We're just observing at this point what's happening. Next. So bioaccumulation of the cyanotoxins in the alewife. On the right-hand side, I just have the formula that we use to try to calculate the bioaccumulation factors. And that's what you'll typically see people talking about when they talk about biodilution versus biomagnification. So when your trophic level 2 is greater than trophic level 1, you'll see biomagnification. When it's less than that, you assume that there's biodilution. So in this particular example, we have something looking at microcystins, and we had something that was less than 1, so that means that it's a biodilution versus the BMAA, which was biomagnification. So some of the things that we don't know, which are very critical, is there is very limited information on what's happening with the anotoxin and whether it is bioaccumulating at all. Never mind whether it's biodiluting or biomagnifying. And we really do not understand the effects of the exposure of these cyanotoxins on fish. Next. And next. And next. Thank you. So these are the results from Lower Mill Pond for 2019 and 2020. For the accumulation of microcystins, which is on the far left-hand side in blue, BMAA, which is in green, and anotoxin, which is in yellow, on Lower Mill Pond. And what you see on the graph, those dotted lines, is the average for both years. And the values there that you see in the upper right-hand corner of each are the toxin concentrations per unit muscle tissue dry weight, which is something that we scientists do. But that's how we all talk to each other about what your values are. So we'll talk about those values in the future. But the interesting thing to note, I think, with all three of these toxins is we did observe them in our juvenile alewife in both years. For the microcystins, generally, we did not see differences across sampling dates, except for one time in October for microcystins. The BMAA stayed fairly consistent across both years for all sampling dates. And for anotoxin, which to me is the most interesting thing, seems to be sort of an outlier, where in 2019, that whole group of the 2019 samples was significantly higher than the 2020 samples. So the question is why? And we might be able to answer that this year. I don't know. But it's very intriguing that we saw such a difference with the anotoxin. Next. So our toxin, when we look at that, so what does that mean for the fish? What does that mean for anyone that's, collecting the fish? So we can look at the toxin in micrograms when it is eaten. So this would be something about what's happening when these juvenile alewife, which are a keystone forage fish for aquatic and terrestrial predators, how much are they consuming when they're eating one of these little tiny alewives that are only 50 millimeters in size. So that's what I've calculated here for both 2019 through 2020. And you can see that there's a significant difference in all toxins, microcystins, BMAA, and the anotoxin from 2019, 2020. The question for me is why? Why did that happen to have a significant difference? And we're just assuming that that's related to what's going on in the pond and the cyanobacteria that are there. And I don't know whether that's a good thing or a bad thing. Next. So what about other fish? Next. So I made up this graph to compare what we found in our study versus other studies in the published literature. Not necessarily for juvenile alewife, because there's very limited information on juvenile alewife. Actually, there is no other information on alewife, but we could compare them to other pelagic benthic feeding fish that people have, gone ahead and quantified the toxin concentration. So our study for microcystins was 10 times lower than what was observed in a study in China. For BMAA, we were pretty close to what they were observing for their concentrations in the muscle tissue. And our anotoxin is actually higher than a study that was published out of Poland. And one of the interesting things that kind of gets me about this whole thing is that these other studies are all working with adult fish specimens that are mature adults, and we are working with juvenile alewives. So obviously the question is this unusual? What's going on? Is there something unique about alewives and their feeding strategy that would give them a higher exposure potential? Or is this something that we might expect that it happens very rapidly and then the alewife basically, hold on to the toxins, it stays within their muscle tissue and they don't depurate, even as they move from the juvenile stage as a freshwater fish out into the marine system. Next. So the things we don't know. Since this study was the first published findings on these juvenile herring muscle tissue, what about other juvenile planktivores or benthic pelagic feeders? We really don't know. We don't have a lot of understanding about that whole thing. At this point, we are just documenting what we're observing. And does this affect their fitness and survival rates? That is a whole other big world of how does this affect these juvenile alewife from these different systems? And we will be continuing to work on that this summer to try to start to just document what's happening at other sites across the Cape. And we will also be working on a project out at Martha's Vineyard that looks at this particular issue. And we have absolutely no idea what kind of effects this has on other aquatic and terrestrial predators. Next slide. So what about Worcester's lakes and ponds? So Worcester has some wonderful lakes that have an incredible diversity of these different types of fish that have different feeding strategies that relate back to their exposure potential to the cyanotoxins. You have your planktivores, your herbivores, planktivory in some part of the life cycle, piscivory as well. So it really, when I was looking at your angler survey, I was like, oh my gosh, look at all of this great, sort of mix of all of these different fish that you guys have. And in my mind, I'm like, well, what's going on with the zooplankton? What's going on with the cyanobacteria? how is this, comparable or different to what we're seeing on the Cape? I'm assuming it would be quite different. Next. So from my perspective, I see that you probably have a very balanced predator-prey relationship going on with your fish populations. And I would anticipate that you would potentially have a low impact on the zooplankton population, which actually is a good thing, because the less amount of impact on zooplankton and the more balanced that population is, the better they are able to process all of this biomass of cyanobacteria potentially, and other algal biomass within the system. So that could mean many different things, but overall, you have a really healthy fish population. One of the other things that I noticed is, Jackie has kind of shared with me some of her data from what you folks have already collected so far. And given what you have for cyanobacterial populations in these systems, you would probably be more concerned with BMAA and Anotoxin, if you are concerned about what's happening with the cyanotoxins in these systems. And we've been able to document that both of these toxins, the BMAA and the Anotoxin, can potentially move through the food web. Next. So I just want to say thanks for being able to share what I consider to be pretty exciting research that we're doing down on the Cape. It's been done as a collaborative effort with UNH. We have some Brewster Ponds Coalition, which is a nonprofit citizen science, organization that's been helping us out. The Association to Preserve Cape Cod, which is also a nonprofit. They do many different things. And then the Massachusetts Department of Marine Fisheries has helped us out as well. So they've been very supportive in doing this work. And I definitely want to thank Jackie for, inviting me to share all of this with you, because it's been really exciting for us and challenging. We're learning a lot, but it's a very important topic for us. And then, of course, Jason for chatting with me about what I've been doing on the research down on the Cape.
Excellent. Thank you so much, Nancy. This was an incredible presentation. And it's such cool research. We do already have some questions that are coming in. So Beth asks, oh, we've got a few coming. So I'll start. Mike asks, well, this one's for Jason, actually. Can you offer an opinion on the loss of the abundant rainbow smelt population that once existed in Lake Quinsigamond in the 70s and 80s? Does it have anything to do with the low oxygen levels and them being forced into stressful water zones? Or could it be stormwater runoff in the streams that has impacted their spawning grounds? Has Mass Fish and Wildlife considered stocking a hardier species like alewife?
Can you hear me?
Yep.
Yeah, so I think the question came from Mike. I say it would be a combination of both those factors. So as you lose, because smelt are obligate cool water species, they need slightly colder temps than your normal warm water fish, and they got squeezed just like trout did. So that was a problem, obviously, and because they require multiple different habitat types to complete their annual life cycle, they need cool water habitat in the main body of the lake, but then they also need that good spawning habitat at the mouth of creeks. And that has been degraded over time as the watersheds feeding Quinsigamond have become more and more developed, and the water quality is deteriorated and sedimentation. So it was kind of a double whammy, so to speak, where they didn't have good reproduction in some years. And then sometimes during the summer, their summer habitat, would decrease, and it eventually resulted in their demise. In Mass Wildlife, I don't know. Rainbow smelt were first stocked in Quinsigamond, or they weren't stocked by us. They were a bait bucket introduction around the turn of the century. I saw a note in an old in an old, it wasn't a publication, it was just a map that was from the 1800s, that someone thought it was this Tom gentleman from Worcester who actually introduced them for the first time. In Mass Wildlife, we don't stock warm water fish anymore. There was a period in the time in the 70s when there was some fish being moved around, and we generally try to avoid doing that because it can have unintended consequences. If you know smelts, the biology of smelt, they can have huge year classes and then also have huge die-offs in any given year. And we have to take that into account when we used to put fish in different places. So we're not currently considering restocking smelt into Quincy.
Thanks, Jason. So for Nancy, a question from Beth. Why are toxins being produced at Mill Pond, and are there some level usually present, or is there a cyanobacteria bloom when it's coming?
That is a great question. So this goes back to my comment about what is happening in those first 10 weeks between when the alewife are hatching and when we are catching them. And the fact that we're seeing cyanotoxins in their muscle tissue at concentrations in July, which are also not significantly different from what we see in their muscle tissue in like either September or October. So comparing the muscle tissue concentrations to what we see for toxin concentrations in the water column, that's where it became very interesting, because we could not correlate those two metrics with each other. And that's where the whole feeding strategy came in. So it was about exposure to potentially not what was in the water column, but what was something that was more concentrated potentially in the sediments themselves, which drove us to collecting sediment cores and quantifying what was in the toxins in the sediment themselves. So yes, throughout the entire season in Lower Mill Pond, in that size fraction that we would expect them to be ingesting, whether it was the Pico fraction or the BFC fraction, we were having toxin concentration all season long. And that's why it became a very interesting sort of world to wander into, to figure out when did they exactly shift their feeding strategy and what were they exposed to, and how would we calculate a bioaccumulation factor for them. But yeah, it's the toxins were there season round in varying concentrations, but their bioaccumulation was not dependent upon what we would consider bloom conditions, which did not happen until late September, October. So the whole world of what's happening with the Pico Cyanobacteria and how and when they are producing the toxins obviously was the driving factor for the accumulation in the alewife. So that's in particular for Lower Mill Pond, which is kind of a unique system. But it would be, we've had it on our minds. We would love to explore other systems that are functioning differently in terms of what might be happening with these pathways.
All right, we have another, it's a more general question. Well, it's related to toxins. Eric asked, so toxin levels will increase as global warming continues?
Potentially, yes. The toxin concentrations that you typically see in these different size fractions can be very much dependent upon how fast they're growing. So the toxins themselves are something that's called a secondary metabolite. So after the cyanobacteria grow and grow really fast, like you had mentioned in your introduction, they all of a sudden they have the ability for whatever reason to grow really fast. That is when they are sort of at the peak of their growth is when you see these secondary metabolites, the toxins being produced within the cells themselves. So yes, as temperature is increasing and cyanobacteria are thermophiles, so they love warm temperatures, you would tend to expect to see that happening.
Excellent. And when there are toxic blooms, how do you control them?
Well, this is a really interesting question. And which goes back to this whole alewife thing and what's happening with the dynamics of the growth of the cyanobacteria themselves within the system. So what we found in Lower Mill Pond is what we call a top-down trophic control of what's happening with cyanobacterial biomass. So from that perspective, if we wanted to control or manage or remediate what was happening in Lower Mill Pond, it may be something more associated with managing the alewife themselves, because they are the ones that are sort of in control of what's happening with the cyanobacterial population via the zooplankton, versus a nutrient management approach, which is what we call a bottom-up approach about nutrient inputs and that being available to the phytoplankton to then grow, which then increases in volume, and then they grow and they increase their toxin production. So I think one of the first things you have to understand is the system, and whether it's something that's a nutrient controlled dynamics, which is the bottom-up approach, versus the top down, and that can help dictate what your remediation strategy could be. If you're doing, if it's a nutrient-based thing, you're looking more at controlling what's going on with nutrient input. If it's a top down, it's more of a biomanipulation strategy that's going to be to give you an effect that you want to see for remediation.
That's really interesting. Jason, how does the anoxic zone come into play in a shallow lake? Indian Lake averages about 10 to 15 feet.
I would ask you, Jacqueline. I mean, what does the anoxic zone look like as it goes through the summer?
So Indian Lake is pretty well mixed. It has a lot of wind action on it. We find that we have pretty good oxygen profiles throughout Indian Lake. At the very, very bottom, we'll have some points during the summer where it will become anoxic, but for the most part, the water column is well mixed and well oxygenated, even as it gets very warm.
Yeah. If a thermocline doesn't set up, if you don't have that temperature density gradient in the water column mixes, then that anoxic zone doesn't form and it becomes a moot point. There are other ponds in the state, where you have a very strong thermal gradient. I'm trying to South Pond in one of the brook fields has a very strong thermocline. It has a big trout layer too. A lot of ponds on the Cape have very strong, wide trout layers, but they do form anoxic zones during the summer. Well, the greatest example I can think of is Quabbin Reservoir. It's 110 feet deep. In every single year, it is oxygenated all the way down to the bottom, which blows my mind.
That's amazing. We actually do see the phenomenon that you were demonstrating in your slides at Lake Quinsigamon very clearly. So great. Any more questions? I have just a few more slides to talk about what we're doing here in Worcester as far as monitoring and treating our cyanobacteria blooms.
So someone was asking about
one more
freshwater.
question. Oh yes, great. Freshwater clams.
Clams. Yes, the freshwater clams. Oh yes, very, very much so. So the whole thing about the bivalves, so the Asian clams are more of an invasive freshwater clams. At UNH, I did some stuff with freshwater clams way back when I did my master's back in the old days. But yes, certainly they do play a role in terms of what's going on with the removal and cycling of the Pico Cyanobacteria, more than necessarily what's going on with something that might be 20 to 30 microns in size. But yeah, whatever's happening with the biotic variables in the system, what they're ingesting, what they digest versus what they don't and they egest as fecal material, plays an enormous role with what's going on with the cyanobacterial population, because they could either be eliminating some parts of it, or just cycling it through, making it available, more bioavailable in terms of internal nutrient recycling. So yes, very much a big part of this whole story about what's happening with the food webs and where is everything going, and then how does it influence what's going on with the nutrients and the toxins themselves. So, and the structure of the cyanobacterial population itself. Yeah, so very much so. Great question.
Excellent. How big a threat is the cyanobacteria for recreation? All right, well, I guess I could talk about that right now.
Yeah, there you go.
Great, great segue. I will jump back into, So I guess I just wanted to talk really quickly about what the city is doing as far as monitoring these blooms, trying to figure out when they're occurring, when there are those certain thresholds that are being met, or indicators that we might have a bloom, and also how we're responding to keep people safe. Because the Lakes and Ponds program, they're primarily interested in, the recreational use of these water bodies, and generally that is when we have high recreational use, we also have a healthy ecology. So we do a lot of monitoring in the lakes and ponds in Worcester at Coes and Indian Lake, which have been identified as lakes that are prone to having blooms. We do measure directly the cell density, or how many cells of cyanobacteria there are. And if it's nearing that threshold that the state identifies as being potentially problematic, we will close the lake for recreation. Or we might treat the lake before it gets to that point, and I'll talk about that in a second. We are also monitoring pigments that are associated with cyanobacteria cells. They use chlorophyll and phycocyanin to absorb that sunlight and turn it into energy, and we could measure those with certain devices. This is a device on Lake Quinsigamond that we deployed, we were testing out last year. It's a solar powered device that's measuring those pigments and sending back that information via cellular signal. At Coes Reservoir, Indian Lake, Bell Pond, and Lake Quinsigamond, we monitor them twice monthly for phosphorus, transparency, temperature, and oxygen. These are all indicators that you've heard about tonight that might tell us when a bloom might be occurring, and just telling us about general lake health. And we will have those closures if we feel that we're reaching a point where the public might be in danger of being in contact with some of these toxins. And we'll measure those toxins directly if we do feel we're getting to that point. We also have the Worcester Cyanobacteria Monitoring Collaborative. This is a group of community scientists that are looking for the presence or absence of certain types of algae and cyanobacteria at 11 or more lakes throughout Worcester. They're actually using methodologies that Nancy created to collect these samples and identify them under the scope. We are planning to expand the program in 2021 to have a little bit more quantification, to do a little bit more with that data, and maybe collaborate with Nancy a little bit more on some of the projects that she's doing to give us more insight on things like food webs and how maybe cyanotoxins are moving through the ecosystems here. So that's all really exciting stuff that we're hoping to move forward with in 2021. As far as the management How are we preventing them from occurring, these cyanobacteria blooms? We work with our sewer division to install green infrastructure. So these are creating opportunities in a space that is very impervious, as Jason was talking about. There's a lot of hard surfaces not allowing infiltration of those nutrients into the soil. We create artificial spaces for that to occur. This is a pretty advanced rain garden that we installed at Shore Park near Indian Lake, I think in 2019. We have plans to install another one this year at Lake Quinsigamon. And basically it filters all the runoff off of specific areas of parking lot or road, and it goes through the soil and filters out, or through the plants and the plantings and the soils will filter out a lot of those nutrients before the water goes into the lake. We do a lot of education on stormwater management, things not to put in the catch basin, you know, like dog poop or your fertilizers from washing your or from fertilizing your lawn or washing your car. All those things will go into the lake eventually, and so letting people understand how those things affect lake health. We also are trying to treat phosphorus before it goes into the lake by applying flocculants to it at places where we know that it's entering in really high quantities, and kind of pulling it out of the water column before it gets to be too much. We manage the lake currently at Indian Lake. We apply aluminum sulfate, that flocculant, to strip that phosphorus out of the water once a year in order to slow down the accumulation of those nutrients. We may have to use preventative algal treatment, so algicides at several points during the year if we see the population getting too high. And then that alum dosing station that I mentioned before, that's just a more effective way of applying those flocculants to the lake. So we are measuring, we are trying to protect public health. There's more that we need to learn. There's more that we can do, and we're moving in that direction. If you want to be part of this, if you're really excited about like what you can do to help contribute to this, our Cyanobacterium Monitoring Collaborative is going to have a training on May 13th. If you join the training, you're going to learn how to use a microscope, take water quality samples, contribute to these like studies that are really, it's like a nationwide study at this point. But also gaining an understanding of like what's going on at any local lake and pond, and any lake and pond could participate in Worcester County. Like it doesn't have to be in Worcester. It doesn't have to be in your backyard. If there is a lake that needs to be studied, if you don't have one, and we can give you one. And you'll get to meet other people who are passionate about this kind of stuff as well. You could sign up for that training on the Lakes and Ponds webpage, which is accessible using this QR code here in the bottom right of the screen. So yeah, so thank you so much for coming out tonight. Thank you to our incredible speakers for joining us and sharing their depth of knowledge with us. This recording will be available on the Lakes and Ponds webpage in the next few days. It will be on YouTube. If you enjoyed it, come to our other angler events. You could go onto wooanglerseries.com to sign up there. And if you're interested in learning more about your local lakes, get in touch with your local watershed association. Indian Lake Watershed Association is active. Lake Quinsigamon Watershed Association is very active, and Tatnick Brook Watershed Association. Those are the three that are collaborating to put on this series. So yeah, with that, thank you so much for coming out, and we hope to see you again soon.